Bismillahirrahmanirrahim, and now this is the March 22 paper 4-2, and we will be discussing this paper, which is of course on the new syllabus, and of course there is no section B in this, which is there are no more essay questions in this. As you can see here, it is for 2 hours, and if you look at the instructions, they'll answer all questions. There's no section A, section B, which was in the previous papers till last year. Starting with the first question. Usually the first question is slightly easier to make you all relax and to let you all get used to the paper and start doing it. The European hedgehog, whatever, I usually don't read this because this unnerves me. The European hedgehog is a small omnivorous mammal. So mammal, omnivorous. Its body is covered in spines which are usually brown. A rare variant which lacks the brown pigment has blonde, pale yellow spines. This characteristic is coded for by a recessive allele. So the ones which are blondes have the recessive allele. So they must be small a, because the other one dominant would be big A, or big A, small a. So just a quick revision that when we were talking about this, you have to think like this. So the brown pigment ones are going to be either big A, or big A, small a. And the recessive ones are going to be always this. So the blonde ones are the blonde yellow spine. This characteristic is coded by a recessive allele. So everybody needs to read this twice. Everybody needs to comprehend it, just like I'm doing it with you all here at this point in time. Figure 1.1 shows a European hedgehog with blonde spines. Maybe this could be a colored picture in the real exam. Part A, Alderney is a small island between the UK and France. Hedgehogs were not found on Alderney until 1960, when 3 pairs of hedgehogs were introduced to the island. The hedgehog started to breed and some of the offspring had blonde spines. Interesting. That means this must have been the case. You see, if this was the case, then it couldn't have been, because if you cross this with big A, then there's no such possibility. But if you cross big A, small a with big A, small a, then you can get these. Now keep on thinking like this, like I am thinking. Three pairs of hedgehogs, so 6 of them. The hedgehog started to breed and some of the offsprings had blonde hairs. By 2017, the population of hedgehogs, now this is we're talking of 1960, so fast forward it another 57 years, 2017, the population of hedgehogs, including individuals with brown spines and individuals with blonde spines, had increased to approximately 600. So originally 6 and now they're 600. Brown and blonde ones. Suggest reasons why the population of hedgehogs increased to such a large number. Now basically, you've got to understand any animal which has increased in large numbers has to be there's ample food for them. There's ample food for them. So the first point is high birth rate, short generation time. They mature very quickly and then they give a lot of they have a lot of offspring. Like if you have a pair of rabbits and within a year you'll have maybe 20 rabbits. So high birth rate or short generation time, then no predators, nothing to eat them. Oh, they've got these spines, they're lethal. You can't even catch them. So no predators, then little competition, plenty of food available, no diseases or fewer diseases. Maybe they don't have, there aren't many diseases and they don't die of any diseases. Like look at the human population, how much COVID-19 has killed so many people. So well adapted and no selection pressures against them. So they have ample food, they don't have any diseases, they have nobody to eat them, they have no predators. A time will come and they'll become a nuisance, because that is what has happened in New Zealand and in Australia with their toad. They introduced a few of them and now there are millions of them and there are no natural predators. So there's become a big problem. So these were the answers that you were expected for 2 marks. Just a quick look of how you would word these. High birth rate, no predators, little competition, plenty of food available, no or fewer diseases, well adapted, or if you said no selection pressures against them. Then coming to part 2 of the question, a survey taken in 2017 showed that 60% of the hedgehogs on Alderney had blonde spines. 60%. Suggest and explain reasons why the proportion of hedgehogs with blonde spines was so high. Well, the main reason is whenever something becomes more popular or becomes more common, It has to be at a selective advantage. Blonde spines have a selective advantage. Maybe they have better DNA, which doesn't make them die of diseases which others die of. Maybe they are camouflaged. Maybe they are better protected from the environment. Maybe the color gives them some advantage. So, but basically, why are we getting this? The basic reason is that there is inbreeding. At the beginning, there was a small gene pool. We only started with 3 pairs of hedgehogs, or these hedgehogs. So there were only 6 of them, and they would only have a certain alleles. There weren't hundreds of them which were introduced. There were just 3 pairs. So the inbreeding means the same keep on breeding, the kids keep on breeding with each other, so that's going to cause inbreeding, ultimately inbreeding depression. Then it's a very small gene pool at the start, or you could have said there's little genetic variation. And of course, all these are founder effect or genetic drift or bottleneck effect. All these words could have been used. That this is, they're all very common words which we use in these situations. And of course, the most important thing, this was directional selection. This was directional selection because one certain type is becoming more common. So if you plot it as a graph, then the graph shifts to the right or to the left, and that is called directional selection. So let's look at the wording. Directional selection, 1 point. Blonde spines have a selective advantage. Allele frequency for blonde spine increases. It is increased homozygous state. Remember, blondes were all recessive, homozygous recessive. Then this results in inbreeding. Why? Because there was a small gene pool at the start. There were only 3 pairs of hedgehogs. So little genetic variation. Like for instance, look at a human example. If only there were 6 people and all of them were blood group O, then A, B, and AB, O, AB, A, group B, and group AB would never exist in that. So small gene pool at start, little genetic variation. If we only had these 6 people living on an island and they were only breeding and they were only having offspring, so then of course that everybody would be blood group O. They would be, because all these 6 people were blood group O. So there would be no A, no B, and no AB. Just giving you a very stupid example, but that's it. Small gene pool at start, little genetic variation, founder effect or genetic drift or bottleneck effect. All these are very similar, and which actually describes how the populations dwindle down and the new population starts from a very few numbers. So that is what it was. Then coming on to question number B. The hedgehogs on Alderney are an example of an invasive alien species. Explain why it is sometimes important to control invasive alien species. Yeah, same thing I was talking about earlier. You have to understand is why invasive alien species can really be a pain in the neck. Why? Because number one, they're going to compete with native species. For what? For food. For the same niche or for the same resources or the food source of the native species. Maybe there are other animals we have the same food source as well. So they're going to compete for the food. Next, they're going to feed on the native species. Then they can cause extinction in the native species. They can breed more successfully than the native species. And often, that is what the case was in the toad as well, which was in Australia. They have no natural predators. And this, of course, would result in disrupting habitats and food webs and food chains, because one of them would become the bad guy. These would become the bad guys, and they would eat up all the food. They would maybe destroy other. They would maybe kill other animals. Maybe they, like in the Australian frog, it was producing a poison that killed all the snakes, all the dogs, because they would squirt out a poison. So causes reduction in biodiversity, or they could maybe introduce a new disease amongst the other animals, because they would all be mixing up in the wild. So there were a lot of answers to this, but you only had to give me 4 of them. So how would we word all this? Compete with native species, 1 mark for food, second mark. Number 3, feed on native species. Number 4, can cause extinction in native species. Number 5, can breed more successfully than native species. Number 6, often have no natural predators, may disrupt habitats or food chains. This will, of course, cause a reduction in biodiversity and may introduce certain diseases. So there are a whole lot of points, but you only had to give me, there are 9 points, but you have to only give me 4 out of these to get your 4 out of 4. Now coming to question number 2, giant axonal neuropathy is a rare autosomal recessive. Autosome means it's on the body chromosome, not on the sex chromosome, which affects neurons. Mitochondria can move freely within the axons of neurons. In GAN, which is giant axonal neuropathy, they've given you the abbreviation at the beginning, the axons of neuron becomes enlarged and blocked by the accumulation of specific proteins. This prevents the free movement of mitochondria and affects the transmission of action potentials along the axon membrane. Number 1, suggest and explain why preventing the free movement of mitochondria when within the cytoplasm of the axon affects the transmission of action potentials along the axon membrane. So imagine something, like you can move about in a room and now you can't move about. Now they're saying mitochondria could move inside an axon and now they can't move inside an axon. So what are the possible suggest and explain? So less ATP for the sodium potassium pump prevents the re-establishment of the resting potential. The sodium potassium pump works because of ATP and then the resting potential is maintained. Also, the action potential, you need that, so reduces the transmission of action potential. So you have got to understand is what is the normal function of the axon. The normal function of the axon is to pump out the sodium potassium pump. So if there is no ATP, then of course we're going to have problems in this. So less ATP for sodium potassium pump reduces the re-establishment of the resting potential. You see, for resting potential to be maintained, the sodium potassium pump has to keep on working. So that will not happen and it reduces the transmission of action potential. That is the purpose of axon, is to transmit action potentials. Part 2 of the question, a nerve condition, nerve conduction velocity. A nerve conduction velocity NCV test can be used to measure the speed of transmission of nerve impulses along neurons in different parts of the body. NCV tests were carried out. NCV is given here. NCV tests were carried out on 3 people with GAN. The speed of transmission of nerve impulses was measured in neurons in 2 nerves. The median nerve, one of the main nerves to the arm, and the peroneal nerve, one of the main nerves to the leg. So one is a nerve in the arm and the one is a nerve in the leg. Table 2.1 shows the NCV test results for the 3 people with GAN and the expected range for a person without GAN. So location of median nerve, where was the median nerve? One was going to the arm, the peroneal nerve was going to the leg. Now person 1, person 2, person 3 expected range. Now and remember these 3 people had GAN. They had this disease. So 47, 30, 43 and the range is 50 to 65. So all of them have less than this. Then in the peroneal nerve, they were 44, 22 and 28 and the range was 40 to 45. Well, this one seems a little okay. And these two are little chungar and they are the ones which are very low. Speed of transmission is in nerve impulses is in meter per second. So person 1, 47, 39, 43 and the range is 50 to 63. So 50 say all of them are less than 50. Here it was 44, 22, 28 but it's 40 to 45. So this one is an okay. These ones are not okay. With reference to table 2.1, describe the effect of GAN on the speed of transmission of nerve impulses. At this data, what do we see? We see decreased speed. You see, all these are decreased. All are decreased speed. And the decreased speed is in both nerves, in the median nerve and the peroneal nerve. So both in the median and the peroneal, or you said in the arm and the leg. So one mark was decreased speed. Number 2 was in both arm and leg. Number 3 was then you give me a data code. If you said 43 meters per second as compared to what? How do we get the average out. You see, you'd calculate the average. So it was 43 meters per second as compared to 31.3 meters per second. So you added all of them and you took out the mean, and the mean was, that's why, 43 meters per second. For this one, it was 31.3. Now let's look at the wording. Decreases speed in both nerves, arm and leg, 43 meters per second versus 31.3 meters per second. And person 1 not affected so much. If you look at this, person 1, this is 44 and this is 47. So this was 50 and 40. So these here he's okay in this one, but here he's not okay in this one. The slight difference there, but these are much lesser than this. So person 1, you must also look at whenever there's some sort of anomalous figures, you must pick that up, because whenever they ask you with reference to table 2.1, describe the effect. So with reference to table, if there's any anomalous reading in that, in which it's something everybody is following a trend and then there's something different in one situation, then you talk about that situation. Now coming to part 3, walking requires nervous control to coordinate movements. One of the first signs of GAN is having problems with walking. Explain how the speed of transmission of nerve impulses in people with GAN can affect their walking. Basically, in walking, what has to do? Muscle has to contract. So it takes longer for muscles to reach the neuromuscular junction or the muscles. And there'll be slower muscle contractions. And the walking problem, what will be the walking problem? They'll be walking slowly, or maybe they stumble, or they trip, or there's reduced muscle control, and there will be slower reflexes. So you have to associate all this with how the transmission of nerve impulses is going to affect the walking of a person. So how would you word it? Takes longer for impulses to reach the neuromuscular junctions of the muscles, slower muscle contractions, reduced muscle control while walking, or you could have said walk slower, or you could have said stumbles or trips, and the last is slower reflexes. Now, let's come to the B part of the question. GAN is caused by a mutation in a gene that codes for a protein known as gigazonin. Scientists have tested gene therapy in mice with GAN. In one study, viral vectors containing a functioning allele of the gene that codes for gigazonin were made. Mice with GAN were treated with one dose of these viral vectors at 12 months of age. 6 months after treatment, when the mice were 18 months old, the scientists used a rotor rod test to measure the effect of the gene therapy. In the rotor rod test, the length of time the mice are able to balance on a moving platform is recorded. The longer the length of time the mice can balance on the moving platform, the better their neuron activity. So it's all totally new situation that you have to read. So you need to slowly read it, you need to understand it, you need to sort of read it again. The rotor rod test was repeated on the same mice each month until the mice were 23 months. So we started off 12 months, 18 months, and then 23 months old. The rotor rod test was also carried out at the same time intervals and ages on mice with GAN that were not treated with gene therapy, and on mice without GAN. So we have three groups, one with GAN getting this, and one without GAN, and one who were normal and weren't having any GAN. All mice were kept in the same conditions, same place, same cages, same food, same temperatures. The results are shown in figure 2.1. Now here comes the test. The graph is the one which is going to test how well you've understood this question or not. Now whenever a graph is given, you always read what is the question and then go back to the graph. Don't, sort of study the graph initially, but then read the question on the graph and then go back to the graph. From the results, some students concluded that giving the mice with GAN one dose of gene therapy had a benefit, but did not cure the mice. Second point, the results of the rotor rod test were not affected by the age of the mice. So the results of the rotor rod test were not affected by the age of the mice. So how did the student conclude this? With reference to figure 2.1, discuss whether each of these two conclusions is justified by the data. Is it justified or is it not justified? So you go back to the graph and then you look at it again. And you say, look at mice without GAN. Let's look at it now carefully. Mice without GAN is this one. Let me just color it just for my own. And then we have mice with GAN plus gene therapy. Now that is this one. So mice with GAN with gene therapy and mice with GAN and no gene therapy. So you have to compare it with this, right? So mice with, so this would be this one, mice with GAN and no gene therapy. Then we have this one, mice with GAN and gene therapy. And then we have mice without GAN. So these are the ones without GAN. So we have got to now see the comparison. First point which comes to my mind is that you look at the one, this is with gene therapy. So the blue is the with gene therapy, right? And the yellow is no gene therapy. So now if you look at the blue line, that's always before that. So they have more balance, they have more, they have a higher balance time. So the ones with gene therapy, so there was a benefit of the gene therapy because they have a higher balance time, or mice with therapy have a higher balance time. Now, why is it not a cure? Why is it not a cure? Because if you look at it, mice with therapy and mice with no therapy, there is a similar balance time at 23 months at the end. There are similar balance time after 23 months at the end. Then, if we talk about the results, are they affected by age? Now, three mice without GN increase balance time during the experiment with age. Here, you see here the point. You see, if you look at the point here, the third point, which is in the results are affected by age, mice without GAN increase balance time. So their balance time has increased. They have no GAN. So mice without GAN increase balance time. Then you gave me any comparative figures. If you gave me that, you got a mark for that. Or you could have also said you need a statistical test to either prove or disprove this. So if you did a statistical test, I think that would be a better idea to really find out what conclusions do you get from this experiment. I have used different colors to explain all this in a manner. So the benefits are in black. Mice with therapy have a higher balance time. Then the fact that it is not a cure, this I've written in red. Mice with therapy have lower balance time as compared to mice without GAN. Then the fact that you said it is affected by age, that's I've given it in green. Balance time decreases for mice with GAN. Then you gave me comparative figures from the table, and then all you need to do a statistical test. Now I'm going to talk about the figures and what comparative figures you've got to give me. There's a whole list of this in the mark scheme, and I've just given you some of them, but they have for everyone, for 16, for 18, 19, 20, 21, 22, and 23. I've just given you a few of them. So at 18 without GAN it was 122 seconds. Yes, you have to give me what was the unit here. So you have to give me seconds here, you have to give me seconds here, you have to give me seconds here. So 18 it was 122, and then 128 and 114. This is without GN, with GN and gene therapy, with GN, no gene therapy. 20, 120, 88, 40, 146, 80, 56, 154, and this is plus minus. This is plus minus 1. So you could have made a little, there could be a little error in it. So it's plus minus 1, it has been allowed. So you just have to look at it again and see how I have figured all this out. Please look at the graph again. And then, of course, all these, you can easily see this. In this video, only question 1 and question 2 have been discussed, and we will continue this in the other videos because these are rather long questions which I want to discuss in great detail. Thank you for watching. Please do subscribe.